Good afternoon, and thank you for joining. I'm Matt Roth with Baylor College of Medicine, and I welcome you to today's presentation. The Extracellular RNA Communication Consortium is an NIH Common Fund program which works to advance the science and research of extracellular RNA. The consortium hosts monthly presentations on a variety of research topics, and I'm pleased to introduce today's speaker, Jim Patton, the Stevenson Professor of Biological Sciences at Vanderbilt University. The title of his presentation is KRAS-dependent sorting of microRNA to exosomes. Jim?
Thank you, Matt, and thank you to Kayla for the two of you for organizing this. I'm happy to give this talk today. Well, as you can see in the title there, and I'm going to talk about microRNA sorting into exosomes. We tend to focus on colorectal cancer and the role of KRAS, but we're trying to do general principles as much as we can. So our U19 group at Vanderbilt is Bob Coffey, Alissa Weaver, myself, Kasey Vickers, and then we have bioinformatics and statistical support from Chi Liu. Overall, the goals of our work are taking off on earlier work that Bob Coffey had done that I'm going to show you about the role of exosomes in stimulating some proliferation and invasiveness in colorectal cancer lines. But then we've expanded, and we would like to know, so how is it that RNAs or specific RNAs come to be secreted? What is the gating mechanism possibly playing a role back at the multivesicular body? How are certain microRNAs destined for export and certain RNAs retained in the cell? Mostly a focus on microRNAs on that front. And we've tended to focus largely on exosomes, and we're doing a sort of a traditional centrifugation, purification of exosomes, and most of what we've done, I'll show you the size range that we're looking at. So most of the time, we're looking at exosomes, but we do know that our exosome pellets also contain RNA protein complexes in them and possibly lipoproteins. And so I'll probably ignore that a little bit, and we'll just say that operationally we talk about an exosome pellet. We're very actively researching right now sort of the heterogeneity in these exosome pellets. Okay, so moving forward, the background of this story is that there's a series of isogenic colorectal cancer cell lines that were generated many years ago. Shirazawa in Japan, they're from the DLD line, which is has one copy of wild-type KRAS and one mutant allele, and then through homologous recombination, they either created a line that's just wild-type or a line that's just mutant. And what you'll see, what we're going to do is to take these three lines and isolate RNAs from the cells and from exosomes, and then do a lot of RNA sequencing. They are mutant G13D mutation in KRAS, and this activating KRAS mutations occur in lots of colon cancers, and you can see the downstream pathways from the EGF receptor through KRAS down to other kinase cascades that then lead to that proliferative effect in these cells. And our goal right off the bat was what is the RNA and the protein composition of the exosomes in these different cell lines, and what's the difference between normal and cancer cells. And going back to some work done in the Coffey lab in 2013, Michelle Demery-Beckler, she had shown that you could take exosomes from mutant KRAS cells and incubate them with wild-type cells, and then you would get an increase in colony number. And also, you could see that there in the KRAS exosomes, there was an increase in KRAS protein and in EGF receptor itself. So mutant KRAS was being transferred to these wild-type cells, and presumably being involved in both growth, and then in the next slide, a matrigel invasion assay where they were able to show that incubation of exosomes that had enriched levels of EGF receptor ligands on the receptors on the surface gave an increase in the number of invading cells, and further it was exosome dependent in other experiments that they did. So exosomes that had a lot of ampiregulin on the surface of their cells, surface of their exosomes, would stimulate both proliferation and invasion. And that was kind of the starting point then for when we submitted our U19 grant, was to then look at, well, okay, the idea here is maybe EGF related, but what are the other components of the exosomes about RNA cargo and other protein cargo that may be carrying this out? So as I said, we have been using sort of the traditional ultracentrifugation purification that I'm showing you here. Most of the time we've been focusing on exosomes. More recently, Alissa Weaver in particular has been focusing on microvesicles. We're doing a little bit of that. Shown here is the protocol, and over on the right are the typical exosomes that we purify from the different cell lines and the size range that we're looking at. So this is, even though we're currently moving forward to do a lot more analysis of the heterogeneity of these pellets, this is what we've been characterizing to this point, from which we did almost all of our sequencing and analysis that I'm going to tell you about now. So we have done a lot of RNA-seq data under many, many different conditions. Shown over here, so originally Jim Higginbotham and Bob Coffey's lab had been using ionomycin to increase, and so we actually had done RNA-seq after treating cells with ionomycin to generate exosomes, and we've done it plus or minus riboreduction and plus or minus serum. So, there we go. And so we have a lot of data where we've now analyzed from short RNA-seq data for microRNAs. We're now in the midst of doing long RNA-seq. I'm sorry, every time I touch my mouse, the slide is advancing. And we've done circular RNA-seq. Beyond that, we've also done proteomics from all of these fractions. And as I said, Alissa is now moving into doing a lot of the, or more, proteomics and analysis of microvesicles. So that's the backdrop of all this. What I'm mostly going to tell you about today is a little bit of the short RNA-seq and the RNAs that we identified. And then we are in the midst of just about ready to finish publication on the long RNAs that we find, things greater than 200 in our exosome preparations. Okay, so ahead of all this, one hypothesis would be that just microRNAs that were highly abundant in the cells would also be highly abundant in exosomes. And that actually turns out to not be true. There's a very distinct export of specific microRNAs out of cells into the exosomes. And what you're looking at on this slide is principal component analysis from our 3 different lines. So if you remember, we have mutant KRAS, a heterozygous, and a homozygous wild type. And what you're looking at here is that when we did microRNA, small RNA sequencing, if you look at the populations in the cells over here on the left, the cell populations cluster fairly close together, are not that different. So the overall microRNA expression patterns in the cells are not that different. But even despite that, you can still tell the difference between wild-type KRAS and mutant KRAS. It's not a huge difference, but it segregates. But if we do principal component analysis on the exosome population, swinging over to the far right, then you can see that there's a much greater difference. And first off, it tells you that the microRNA profiles are completely different from the cellular RNA profiles. It tells you that there's some sort of a gating mechanism, some sort of a export control mechanism that only certain microRNAs are exported out of these cells. It's not just a reflection of the levels in the host or in the parent cell line. And secondly, you can see that there's a much greater difference in our ability to distinguish between wild-type KRAS and mutant KRAS when we look at the microRNA profiles. Okay, so moving forward, this is where we're really focusing our efforts now, are what might control, from the microRNA standpoint, which ones are destined for export, which ones are destined to be retained in the cell. Okay, so carrying off on this analysis, we went through all of the RNA-seq data and we identified microRNAs that changed, and we found that there were common microRNAs between the cells and between the exosomes, and then different RNAs that would segregate between whether you were wild type or mutant KRAS, and did all of the differential expression analysis. And I'm just going to focus down here in the bottom of this figure as some examples, and I'll really focus here on this miR100. So miR100 was one of the microRNAs that we found that was enriched in exosomes in mutant KRAS cells, and in contrast, miR10 was enriched in mutant KRAS cells. It was enriched in the cells. So these were RNAs that would show different patterns. One was exported and one was retained. And then we have the miR320 family, which was exported into exosomes regardless of KRAS status. And so we have lots of RNAs that we found that were differentially expressed that we've been tracking. We picked these three because of the patterns that they show between retention and export from wild type to mutant KRAS. One that's not shown here, but it is exported in the mutant KRAS cells, is miR125B. It wasn't as enriched as miR100, but I'm going to come back and tell you a story towards the end of this seminar about miR125. Okay. So knowing that these RNAs are secreted always comes down to then, is it functionally relevant when they're out in the exosomes, exported into exosomes? Are the exosomes taken up by recipient cells, and do they have an effect on gene expression in the recipient cells? So Diana Cha and Jeff Franklin, this work right here that I'm showing you is largely from Diana Cha, set up a series of experiments where we used transwell cultures where we're going to have a donor cell population on top of a membrane and then a recipient cell population below the membrane, so that if there's any sort of transfer of extracellular vesicles that are taken up in the recipient cells, we could monitor this. That would be coming from the donor cells. And the first set of experiments used a luciferase reporter that had known microRNA sites. So if we're using mutant KRAS cells, we're going to put miR100 sites downstream of luciferase. We're going to see if miR100 goes from the mutant cells, donor cells, to the wild-type recipient cells, matching what we found in the RNA-seq data. And this is an example of that experiment done here to show you that when we set this up, miR100 indeed was transferred, and it would silence the reporter. So I'm sorry, my pointer keeps doing this. So when we have perfect miR100 sites, miR100 was exported, and it would lead to this repression in the recipient cells. Okay. To try to further prove that that was due to expression of miR100 in these donor cells, we treated the donor cells with an antagomer. The idea being that if we reduced the levels of miR100 in these donor cells, then we should not see as much repression. And what was shown is exactly what we found was the difference between, we were able to, the antagomer showed that the effect was due to release of miR100 from the donor cells and not some sort of unusual upregulation of let's say, miR100 in the recipient cells. We also did a lot of quantitative PCR, and we were able to show that we got about a 34% increase in miR100 in these recipient cells when incubated in this transwell from the donor cells. So that was with a luciferase reporter that had perfect sites, but then we wanted to find out, could it also then regulate an endogenous transcript? And so for that, we searched through databases to identify targets for miR100, and one of the targets is mTOR. So instead of having perfect sites here, we have the luciferase construct with mTOR at the 3' UTR. And then we went back and did our transwell assay the same way, and we're going to see whether miR100 being transferred from the donor cells to the recipient cells is going to silence mTOR. And as you see here, we saw suppression of that when we have mTOR. In the controls, there's no statistical difference. When we have the mTOR 3' UTR, we saw a miR100 dependent repression of luciferase activity or suppression of luciferase activity in the cells. Okay. So from the antagomers and endogenous and the other reporter assays, the data were fairly consistent that miR100 is secreted by mutant KRAS donor cells and can be taken up by recipient cells. In this case, we've always used the wild-type recipient cells, but it could be taken up by any recipient cell and then silence these reporters. We've done this now for a number of other microRNAs, and I've only decided to talk today about the miR100. Okay, so I'm going to switch gears and talk about, now that data was all published in eLife, the microRNA data. This data that I'm now going to talk about for the next few slides is our continuing analysis to look at the exosome RNA populations and now focus on long non-coding RNAs. And over here on the left, what you can see is that when we do the sequencing and we map the reads back, one of the striking findings is that a lot of the reads that we find in exosomes do not have a perfect match to the human genome when we map them back. They have greater than 2 mismatches, okay? Now, we've done a lot of work to show that these actually are RNAs derived from human cells, so it's not some sort of a contaminant in there. And what we think is happening is that a lot of the RNA that's exported out into exosomes has base modifications or other changes that when the libraries are made for RNA sequencing, there's misincorporation of bases. And so if we allowed more mismatches, we would see that these reads would tumble back towards the expected pattern. But that's sort of an interesting point, is to think about why are there so many RNAs that are base modified that are heading out into exosomes. We actually did some experiments in a collaboration with Peter Dedon's lab at MIT, and he did some mass spec of the modified nucleotides, and there is an incredible amount of modified RNA that's exported into exosomes. And we're following up on that right now to see, is that are microRNA dependent, or are all these modified bases in tRNA fragments, or where exactly are they coming from? But it's an interesting point, I think. Now, that's the overall reads. If we go over and look at the kinds of RNAs that we find in the long RNA-seq, you'll see that most of the reads that we get come from protein coding in red that you see here, so a lot of them are red. A couple of the ones that seem to be most different when we compare, for instance, this is a triplicate of exosomes versus cells. When we compare, you'll see that there's an increase in transcripts derived from pseudogenes, and there's a little bit of an increase in transcripts derived from antisense transcripts as well. So through all of these, as we're going through, we've now gone through the same sort of cluster analysis to identify all the different RNAs and what are unique to mutant KRAS cells to wild-type cells. So on the same principal component analysis that I showed you a few minutes ago for the microRNA data, we've now done the same thing for the long RNA, and that's the Venn diagrams that you see over here. But I'm going to focus over here on this principal component analysis. So in this case, if we do the reads for all of them, you'll see that between the 3 different cell lines that we're using, the long RNA-seq profiles are almost indistinguishable between the cells. You can't distinguish mutant KRAS from wild-type KRAS, and the expression patterns, which probably isn't surprising, these are isogenic cells, are very, very similar. When we do the long RNA-seq in exosomes, you can again see that there is a distinct difference in the RNAs that are exported to exosomes. So it's not, again, the RNAs that are exported are not simply a reflection of the cells, and that holds now for, I showed you microRNAs, and now for the longer RNAs. In the long RNA from mutant KRAS to wild-type KRAS, where I showed you on the microRNAs where you could easily distinguish the cell types, we really can't do that on the long RNA-seq or principal component analysis doesn't show that for long RNA-seq between wild-type and mutant KRAS. And I'll also just note that we do see a fair amount of variability. That's this that you're seeing here from prep to prep. We've done triplicates, but we get more variability in our long RNA-seq reads and library sequencing than we got from the microRNA data. Nonetheless, as we go through all of that data, we could identify transcripts that are differentially enriched from in exosomes to cells and in between mutant KRAS and wild-type KRAS. And I'm just going to show you a few of the cells that are, of the transcripts that are enriched in this process. So this is work that Scott Hinger has done, and he's found RNAs that are upregulated, either comparing cells to cells or comparing exosomes to exosomes or comparing exosomes to cells. I'll tend to focus over here, but so we found RNAs that are upregulated. I'm going to talk a little bit about Rab13 in a minute. So there's mRNAs that are upregulated, and there are long non-coding RNAs that are upregulated when we compare between the different cell types. And we're now engaged in studies to try to understand or try to demonstrate whether these RNAs that we see the reads from, are they intact? And I'll just digress for a minute, and that's something to always be aware of when we analyze reads from RNA-seq. We'll often find reads across the length of an mRNA, let's say, that would almost lead you to believe that that mRNA is intact in the exosome because of the read depth across the whole transcript. You don't have peaks as though there might be sort of stable degradation fragments around. We see a more consistent pattern for some messenger RNAs. And even for those messenger RNAs, though, we have a hard time demonstrating that full-length intact RNA, long RNAs, mRNAs, are present in the exosomes. And that may be us. Maybe we're having experimental difficulties or technical challenges to do the longer intact RT-PCR reactions, or it may reflect that a lot of the RNA that was deposited in the exosome is degraded or is in the process of being degraded. So RNA-seq can fool you a little bit on when you look at the read depth across transcripts of thinking that, well, that's probably intact, and sometimes it may be and other times not. Okay. So we have struggled with this a little bit. We're still working on it. I'm going to show you a couple of examples of how we are testing whether long RNAs can functionally transfer, again, using our transwell system from donor cells to recipient cells. The first that I'm going to tell you is over where Scott has done experiments where I showed you that Rab13 was one of the upregulated ones in exosomes. So he created a construct where we put an HA tag on Rab13 and transfected this into the donor cells, and then the idea is to find out whether the recipient cells have the HA tag Rab13. The recipient cells are not expressing a tagged version, so that when we do the assay, can we show whether Rab13 messenger RNA transfers from the donor cells to the recipient cell. And then I'll also show you how we're using sort of a novel application of a CRISPR display technique that John Wren's lab devised as a tool to try to track other non-coding RNAs that may be trafficking. So on the Rab13 experiment, here are the data that if we do PCR in cells as expected, we can find things, controls like actin was thought to be the control, Rab13, and then BMI1. But if we look at exosomes, we were actually able to detect full-length Rab13 in exosomes, and that these were being transferred. And also, interestingly, actin was being transferred. We never were able to show that BMI. So these are transwell assays where we've looked to see whether the RNA is being transferred. And at least for these RNAs, or at least for Rab13, it appears that it's transferred. Now, I will say that this has been there's variability in this experiment about how often this actually works. So I think it's in line with what a lot of people are seeing, that the levels of intact messenger RNA that move from into exosomes and stay intact and move from cell to cell are, at least in our hands, a little bit variable. We can detect it occasionally. It's not as robust, and certainly not at all like microRNAs, where we have much better handle on which microRNAs move and the fact that they can affect gene expression in the recipient cell. Now, part of the reason that we can demonstrate, detect that microRNAs affect gene expression in the recipient cells is that we have a better handle that microRNAs, we know what they do. They bind to the 3' UTR primarily, and they repress gene expression of that target messenger RNA. For messenger RNAs, that would be easy to do, where we can show the messenger RNA is being transferred and can be expressed, tagged messenger RNA in the recipient cell. For a lot of the long non-coding RNAs that we're interested now to find out whether they may have a role, a challenge is what assay do you use to demonstrate that if they do arrive in the recipient cell, how do you know that they're actually causing some change in gene expression? What reporter assay, what can you use? So to try to address that issue, we had the idea that we could use a technique, a strategy called CRISPR display. So you're looking at this, and I'll explain it here in a little fair amount of detail here. So what we're doing is in donor cells, we're expressing a guide RNA. And in the recipient cells, we have a Cas9 that's fused to a transcription activator, BP64, and then, so it's not going to cleave DNA, it's going to deliver basically a transcription factor, and the Cas9 is going to be delivered to a normalized reporter and then a reporter that does luciferase. So the idea is that if the guide RNA is transferred from the donor cells to the recipient cells, the guide RNA will direct Cas9 to bind to these reporters on our, to the promoters of these reporters and activate luciferase. And this luciferase is secreted, so we can just measure luciferase activity in the media. Okay. This guide RNA that you see here, and now I've blown it up down below, so this is the guide RNA with this loop region that Jennifer Doudna lab discovered for the class 2 Cas9. You can, from the RIN lab, you can insert any RNA of your interest either in this engineered loop that I'm showing you here, 2, or you could add them at the 5' end, or you could add them at the 3' end. Now the RIN lab set this up that they could deliver long non-coding RNAs to anywhere in the genome by targeting, putting these long non-coding RNAs on the guide RNA, and then they would know where they traffic, and they were more interested in would they repress adjacent chromatin. In our case, we want to know if we separate them, express the guide RNA in one cell, all these reporters and the modified Cas9 in the recipient cell, can we see movement of this guide RNA into the recipient cells? Okay, and since we know that miR100 is transferred from our mutant KRAS cells, an idea would be to first off see whether the guide RNA will move by itself, and if not, if we insert miR100 into this engineered loop 2, will it go? Okay, so as I've shown here, if the guide RNA doesn't traffic from the donor to the recipient cell, then you'll get no expression of the reporter. If the guide RNA arrives in the recipient cell and is able to target Cas9 to the reporter, then you'll get reporter expression. Okay, this is from the RIN lab showing that they attached a long non-coding RNA to the 3' end. We actually put our RNAs in this loop that's where my cursor is right here. Okay, so when we did that experiment, we tried miR100, and then we picked another long non-coding RNA that we'd found from our sequencing data sets, and then the controls. And so what you're looking at here is, if you remember, this is wild type and mutant KRAS cells. What you're looking at here is the luciferase activity in the recipient cells, or in the media, actually, and so it had to have been transferred into the recipient cells to give activity. When we put miR100 in there, we can get about a 2-fold increase in luciferase activity. So this is demonstrating that that guide RNA, which is about, I think it's 113 plus the miR100 sequence, so about 130 or so nucleotides is being transferred from donor to recipient. If we put a longer non-coding RNA in there, this is the CRNDE, it's about a 700 nucleotide long non-coding RNA, that basically isn't transferring, and this is the no guide RNA. So this is the, and it's normalized, so this is basically no transfer of the guide RNA. And that's the point of this assay, is we are not seeing any transfer of the guide RNA unless we add an RNA element that endows it the capability to transfer from donor to recipient cells. And fortunately for us, we knew that miR100 is expressed in the mutant KRAS cells, and those are the cells that we see the transfer. We know that miR100 is really not secreted to any great extent in the wild-type KRAS cells, and that comes out as basically background. So the background levels from no guide RNA are fairly low, and then we're getting the same behavior that we expect from our RNA-seq data on miR100 only being exported out in mutant KRAS cells. So it appears that this could be a useful system. I'm going to back up a slide here. We are now measuring luciferase, but moving forward, we have these other markers in there that we'll be able to tell, so that we're also in the midst of creating stable lines that express the different players. The experiment that you're seeing at twofold was done with transient transfection, so I think it's actually an underestimate of the level of transfer that we're getting. So I think this could be a valuable system to now winnow down what are the sequence elements within miR100 or any microRNA that are the export signal for moving a longer RNA, a guide RNA, from the donor to the recipient cells. Okay, so now I'm going to switch gears and I'm going to tell you about a paper that was just a couple weeks ago accepted in Nature Medicine, and this is work done by Wenwen Liu and Xiaodi Zhao that were in the Coffey lab, and this is part of the Vanderbilt Consortium here. And it's coming back to our friend miR100. And in this case, it's that miR100 and miR125 mediate cetuximab resistance via Wnt beta-catenin signaling. So I'll explain this as we move forward. Okay, so in colon cancer, APC mutations, every time I touch the mouse, it moves. APC mutations underlie, and then RAS mutations come along, and then you develop colon cancer. And one of the treatments for patients with colon cancer is to treat with a monoclonal antibody against the EGF receptor. And that blocks downstream RAS signaling. Okay, so cetuximab is this antibody against the EGF receptor. We get this block in RAS. Okay, so the idea here is that in many colorectal cancer patients that start taking cetuximab and it binds to the EGF receptor, they develop mutations that then block the action of this of cetuximab, and so they develop resistance due to mutations. And most of the time it's been thought to be an EGF receptor, but it could also be in downstream signals. And then backing up a slide, if you have mutations in KRAS, you don't prescribe cetuximab because KRAS will be active, and so it's a contraindication for that. So the idea here is that could there be other non-mutational forms of anti-EGF therapy? Okay, so other than mutations in the EGF receptor or downstream RAS signaling, are there other mechanisms of resistance? And to cut the story short, which I'll walk you through now, is that it looks like in cells that dramatically upregulate miR100 and miR125, they're able to resist or develop resistance to cetuximab, anti-EGF receptor. Okay, so that work was done by Ramona Graves-Deal in Bob's lab. Started this assay by doing a very interesting set of experiments where she took HCA7 cells, which normally in 3D will give these what we call a CC morphology. It's a cyst with a central lumen and a polarized epithelium. And these cells in 3D are sensitive and die when exposed to cetuximab. But she did an iterative round of selections going between 2D and 3D and 2D and 3D, and eventually was able to isolate a line that in 3D was now resistant to cetuximab, so CC, cetuximab resistant. These cells grow as you see here without a lumen, and we come over here and on the far right, if you look at this, here's the CC cells with these the cyst with the central lumen polarized lining, and then the resistant cells have a completely different phenotype, and they proliferate in there. So shown over here is the colony number when they become cetuximab resistant. So after, I think it was 18 days, I believe, of this iterative selection, she was able to derive these cetuximab resistant cells. And when they take those cells and actually put them into mice, excuse me, you can see that the cetuximab resistance continues on. I'm sorry, my mouse does this every time. So the idea here now is what are the gene expression changes in these cetuximab-resistant cells compared to the original CC cells that then allows them to be resistant to anti-EGF receptor antibodies. So we did, again, a lot of RNA-seq, long and short RNA-seq, and also, first off, we did a lot of RNA-seq and whole exome sequencing of the cells to show that there were no other known mutations. So no genetic events were detected that could explain the cetuximab resistance. And so it appeared that there was some sort of a novel mechanism of resistance going on. And when all the analysis was done, then we found lots of mRNAs and microRNAs that were upregulated and downregulated. And over here on the you can see that this RNA called miR100HG and these microRNAs that you see here were the most upregulated RNAs when these cells were put into 3D, okay? So the CCCR cells grown in 3D show resistance to cetuximab, and these RNAs are the most upregulated. And interestingly, they come from a host gene called miR100HG, okay? And so here's the gene. It's actually transcribed from right to left that you see here. It's a very long 115 kb original transcript with 4 exons in the host non-coding RNA. So that's this miR100HG long non-coding RNA. After splicing, it's about 3 kb. And then out of this third intron, these 3 microRNAs, our friend miR100, miR125, which I told you early on in this talk was also, we find that in the KRAS exosomes, and then LET7. So these 3 microRNAs are processed out, and then this one. This miR100HG has been published previously by the Emmerich lab that it can act as an oncogene in acute megakaryoblast leukemia. So the question became, in these resistant cells, what role is being played by these RNAs, and can we dissect this out? So first off, we were able to show that not only by RNA-seq, but by other means, this miR100HG, the miR100, and the miR125 are all dramatically upregulated in these cetuximab-resistant cells. If you go over here to the right and we're doing RNA fish, you can see that the miR125 in red is highly upregulated and the miR100 in green is highly upregulated, so by fish and by PCR. If we take a battery of colorectal cell lines, some of which are cetuximab resistant over here on the right and some of which are sensitive on the left, if we look at the levels of miR100HG, this is the long non-coding RNA, versus the 2 other, the microRNAs, you can see that the resistant cells have increased expression. And it wasn't just colorectal cells, that we also found it in head and neck squamous cell carcinomas. So to dissect further about the role of miR100 and miR125, the short story here is that both of these RNAs seem to work together to confer cetuximab resistance. Now, we still are working really hard right now to find out what the relative contribution is of the miR100HG long non-coding RNA, the 3 kb long non-coding RNA, and we don't know that answer right now, but for 125 and 100, we did a series of experiments where we either knocked down the RNA using sponges, or we overexpressed the RNAs, either by themselves or together in sort of a bicistronic expression plasmid. The point here is that if we block expression, let's just focus on the bicistron, okay, they're cetuximab resistant, but if we increase expression of the sponge, meaning decrease the expression of 100 and 125, we get fewer colony numbers. Okay, and you can even see this over here in these CCCR cells. As we decrease the concentration of 100 and 125, they begin to look more and more like the parental CC cells as opposed to the pretreatment CCCR cells. And the same thing can happen in the CC cells, where if we overexpress both of these RNAs in the bicistron, we'll see this increase in colony numbers, so increased resistance to cetuximab, and then again the cells start to look like the CCCR cells. Okay, and it's not just our these HCA7 cells; it happens in 2 other cell lines as well. Okay, so it looks like these 2 RNAs, or microRNAs, are both hitting targets together to confer cetuximab resistance. And it's not only in the colonies, but also then when we do subcutaneous xenografts, it's the same exact thing when we look at this. When we take these xenografts, if we go over here to the right, if we block expression in the cells before they're injected into the mice, you'll get less tumor and in the CCCR cells, and if we overexpress it, we can make the CC cells now become have a higher tumor weight. Okay, so it's consistent again both in the cultured cells and in xenografts that these two microRNAs are conferring cetuximab resistance. So what do they do? So Xiaodi and Wenwen did a lot of work to try to identify targets of miR-100 and miR-125, and what they focused in on was that there are binding sites for miR-100 and miR-125 in the 3' UTR of several genes, but these 5 are all Wnt signaling negative regulators. So they created luciferase constructs where they could check for whether miR-100 would suppress or block expression of a reporter, that DKK1, 3, and as you see here. And what you found is that it held in all cases that these microRNAs, DKK1, can suppress, and if we make a mutation in this site, it won't suppress. And so for DKK1, and the same story for ZNRF3. And as we walk through the whole, all these different RNAs, it looks like these miR-100 and miR-125 bind to and suppress negative regulators of Wnt signaling. Okay? And so then opposite of that is that then it should show an activation of Wnt signaling, which is what we see when we either look at beta-catenin levels, or when we pick out genes that are known to be Wnt target activators, we see increases in these resistant cells. So the idea is that these microRNAs are targeting negative regulators of Wnt signaling, which thereby increases Wnt signaling in the resistant cells. Okay, then besides negative regulators of Wnt, miR-125 was predicted to target GATA6. And this was interesting because if we look here in the middle, there are multiple binding sites for GATA6 in the promoter for the miR-100HG locus. So Wenwen and Xiaodi did a lot of experiments to show that, number one, over here in the resistant cells, there's less GATA6. So miR-125 is up. It represses GATA6. That's consistent here where we see less GATA6 protein. If we increase the concentration of GATA6, you'll see decreased luciferase activity. If we come over here and look at the analyses in the different cell lines, especially if we go through these different ways, it appeared that number 2 was the site number 2 was played the greatest role in mediating this. So miR-125 blocks or inhibits GATA6, which then is normally a repressor of the miR-100HG, so it upregulates miR-100HG. Here's a reporter assay to show that that miR-125B actually can target 3' UTR, and when you have miR-125, you get a decreased level of GATA6. If we use our sponge to block the production of miR-125, then compared to the control, we see an increase in GATA6. Okay. Then a nice set of experiments using the TCGA database, and especially I'm just going to focus over here on they did the lower quartile. So there's a correlation between those that have high miR-100 have low GATA6 and vice versa. Okay. So the data are consistent that there seems to be a regulatory circuit between GATA6 and miR-100HG, GATA6 being a transcriptional repressor of miR-100HG. Okay, and then to show that this would have meaning, several patient tumor specimens were obtained, and then they were looked at prior to the start of cetuximab and then at the time of tumor progression. And what you can see here from pre and post, we see generally an increase in 100, not in all cases, but in most cases, and generally an increase in miR-125, okay? And as those two increase and they block the negative regulators of Wnt, then we see mostly an increase in beta-catenin levels. So, and then over here on the GATA6, a decrease in GATA6. And down here are a series of sections across those tumors where you can see as we go, when the tumor starts to progress, we start to see an increase in the miR-100HG. We start to see an increase in the microRNAs that are encoded by miR-100HG in 3 different specimens that you see here. If we look at beta-catenin levels, there's a dramatic upregulation of beta-catenin, and GATA6 levels are decreased. If we just focus here on case 1. Okay. So all of that led to this a very nice model whereby in cetuximab resistance, this miR-100HG locus is dramatically upregulated. It generates a long non-coding RNA called miR-100HG, but then within that long non-coding RNA, miR-100 and miR-125 are expressed. These two microRNAs target at least 5 negative regulators of Wnt, and that upregulates Wnt signaling, leading to cetuximab resistance. And then to complete the loop, miR-125 represses GATA6. GATA6 is normally a suppressor or repressor of miR-100HG. So then that's what gives the transcriptional upregulation. Okay. So conclusions. Exosomes contain distinct microRNA profiles from the parental cell. I think that's a fairly well-known finding now in the consortium. There seems to be some, or there has to be some sort of a gating mechanism. Both exosomal and cellular microRNA content changes depending on the KRAS status. miR-100 is secreted in exosomes from mutant KRAS cells, but not from wild type, and maybe that will turn out to be a potential biomarker. miR-100 can be transferred. We can see functional effects of that. From the long non-coding RNAs, we're still working hard to identify or to demonstrate transfer of RNAs. But related to then the miR-100 story that we show in the exosomes, we've now shown that in the cetuximab-resistant cells, we see a dramatic upregulation of miR-100 and 125. Now, what I didn't tell you is that we find these RNAs in the exosomes from those resistant cells. So an intriguing possibility that we're starting to work on now is, could this be a mechanism of tumor microenvironment transfer of these microRNAs to increase then the size of the tumor by increasing resistance to cetuximab through exosomal transfer of miR-100 and miR-125. Lastly, and a key part that we're having to move forward now, I'd really like to understand what is the role of the miR-100HG spliced 3 kb long non-coding RNA. We know that it's highly upregulated. What role does it play? And we don't know. So last slide is to give some acknowledgments. So a lot of the microRNA work was done by Diana Cha and Jeff Franklin. Scott is doing a lot of the long non-coding RNA. Bob Coffey's lab, I guess I didn't put them in there, but Wen and Xiao Di did all of the majority of the work on the miR-100HG. And then from the Weaver lab, I didn't talk about the experiments we've done looking at the gating mechanism for now, and I didn't have a chance to talk more about what we're doing for looking at some of the heterogeneity. But other than that, I'll stop and take questions. Thank you.
Jim, very nice. Thank you. Jim's happy to take questions now. Just a reminder to, keep your devices muted if you're not speaking, and then unmute your phone if you have a question. So questions, please, for Jim.
Hi, Jim. Yeah, that was a great talk. So I was a little, it wasn't clear how you envision it within a tumor. Some of the cells become resistant to cetuximab, and so they make more of these microRNAs, and then they release them, and that confers resistance on the other cells, the tumor cells?
Clearly a hypothesis that we don't have any proof. What we know is that all 3 of these microRNAs we can find in our exosomes, miR-100HG, the long non-coding RNA, miR-100 and miR-125, they're in exosomes. Now, on the cetuximab resistance, we know that resistant cells have high levels of these RNAs, and so the hypothesis could be is that these RNAs could also be released to adjacent cells and then confer resistance on adjacent cells as the tumor microenvironment changes adjacent cells.
And what triggers the upregulation of this gene again? Does it encode both of the?
Yeah, we don't really know that. We know that GATA6 as a transcriptional regulator plays a role in this, but what are the other mechanisms for why? And remember, or I didn't, if you could see from the expression analysis, they are dramatically upregulated depending on who's doing the assays. In some cases, it's from 100 to 500-600-fold increase in transcription of the miR100 HG locus. We don't really know what the initiator is to change that gene expression. That'll be a line of work that we're heading on now.
Other questions for Jim?
Hi, can I ask a very sort of naive, non-cancer person question? Like, are there many systems in which there's evidence that chemo-resistant cells confer that resistance to sensitive cells?
Well, yeah, so I'm not a cancer person either. I'm an RNA biologist, so I'm probably not the best person to answer that. I don't know offhand. I guess where I'm making the leap, and you could call it that, a leap, is that we know that these RNAs are present and abundant in the exosomes from these cells. So even though within that cell they're mediating changes in Wnt and upregulate or endowing resistance or allowing resistance to cetuximab, it's sort of an intriguing idea. Well, if these exosomes are laden with those same microRNAs, what's happening if those microRNAs are delivered in the exosomes to adjacent cells? Do they also then upregulate Wnt, and could they become sensitive? I don't know about other models, to be honest.
There are a bunch of exosome world papers about transferring resistance via exosomes. There's a number of reports out there.
Thanks, Jeff.
Jim, I'll ask one. You kind of at the very outset, like obviously you're studying prostate cancer here. Is any other studies in terms of KRAS involvement with EVs and other cancers?
There's a mouse paper concerning KRAS and pancreatic cancer as a secreted marker. That's one I know about.
Okay, thanks. Okay, great. Well, Jim, thank you very much. That was a great talk.
All right.
If anyone else has questions, certainly can, I'm sure, email Jim.